Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And so far what we have discussed about different processes like what are you should follow and we should not follow while you are doing the gel filtration chromatography. To explain you this in a more detail, we have prepared a demo sessions where we are going to take you to my lab and where one of my student is going to show you how to pack this column, how to pack the gel filtration column, how to determine the molecular weight using the different types of approaches what we are going to discuss in a subsequent slides, and how you can actually exploit the or detect the different types of problems in the gel filtration column, such as how to detect the air bubbles, how to detect the air channels and all other kind of problems, and what if you are getting some kind of problems, how to troubleshoot those problems in the column packaging itself.
In this video, we will demonstrate how to perform gel filtration chromatography or size exclusion chromatography. There are various methods are available in chromatography to separate different types of biomolecules. For example, if you want to separate based on size or shape, it is called as gel filtration chromatography, which suits the most. If you want to separate the molecules based on their charge, then you can go for the ion exchange chromatography. So these are various methods are available, but in this video we are mainly focusing on the gel filtration or size exclusion chromatography. What is gel filtration chromatography? There is two phases in this process. One is stationary phase, another one is mobile phase. Stationary phase mainly a matrix, cross-linked matrix. For example, we can use dextran, or another name of dextran is Sepadex. This is highly cross-linked glucose molecules. Or we can use agarose. This is also cross-linked. Or we can use polyacrylamides. But in this video, we are showing Sepadex G75. This is the stationary phase we are using. So in this matrix, it contains beads which having small pores. So if you want to separate a mixture of molecules starting from 1 kDa to suppose 200 kDa. So the small molecule which is 1 kDa, it will permeate through or diffuse into the pores in the beads, and the bigger molecule having 200 kDa, it will excluded from the retaining in that portion of pores. So it will elute first and the smaller molecule will retain there, and we have to give sufficient buffer to elute that one. So this is the overall concept of the gel filtration chromatography. It can be widely used as used in separation of proteins, peptides, or oligonucleotides. So in this video we will show you how to pack the column first and what are the buffers required. For gel filtration chromatography, there are two kinds of columns available. One is pre-packed columns and another one is column material, which can be used for the packing of the column. Here we will show you how to pack a column, and also we have pre-packed column. At the bottom of the column there should be a sintered filter in pre-packed columns, which will forbid going of the beads through outlet. So here if you see in pre-packed columns, there is some sintered filter bottom of the column in both the ways. Outlet and inlet both contains the sintered filters. Inlet, which is mainly from buffer inlet, if there is any particles which will obstruct the flow, they will be separated on top. And also the outlet one, which can be used preventing passage of the beads through outlet. So here we don't have exactly syntax filter, but we can use a piece of cotton. So just put the cotton. So now we inserted the cotton. These are the beads we are going to use for packing up the column. These are Sebadex 75 beads. So there are different materials available: sucralose, Sebadex, Separose. These all are derivatives of a carbohydrate material. So here what we will do, this is a overnight swollen beads. So you have to take the beads complete pack and incubate, soak it in the water or the buffer. So these are swollen one. Now after inserting the this interfilter or column, you have to wash column properly, at least 2 to 3 column volumes with the distilled water. So if anything it contains like dust or any other contaminating particles, it will remove. Now we directly pour the beads on top of the column. If you observe closely, you can see the settling of the columns, settling of the beads. As we can see, the beads are settling slowly. So after complete settling of the beads, then we will put some filtered kind of thing or some piece of cotton on top of the beads. Then we will load the sample. As we can see, now the packing is over. Now what we have to do is we have to equilibrate the column with the 0.05 molar phosphate buffer. So we'll just add phosphate buffer. And drain the any unbound solvent. So here, after equilibration of the column, we will load the sample. After column packing, you have to check the efficiency of the column. So for every column, the parameters V0, that is void volume, and VT, total volume, and V, elution volume, it differs. So for checking of the column efficiency, we have to use 0.2% of total volume of the column acetone we will load. So we have to observe the elution at 280 nanometers. From this we can calculate number of theoretical plates n. So the maximum number of the theoretical plate means the more the efficiency. For calculation of any unknown proteins molecular weight, we should know what is the VT, V0 and VE. VT is the total volume of the column, means the buffer occupied in the spaces of beads and also the buffer in between the beads. So that that will be total volume. The void volume is the buffer in between the beads. Elution volume is uh, where the elute. Suppose we are eluting protein. So at what volume it is eluting, that is called as elution volume. So for estimating the void volume, we can use blue dextran. So first we have to cover top with the piece of cotton, then we will load the blue dextran. After loading, sample loading, we will start collecting the buffer till blue dextran completely eluted. That will give the void volume. So now we loaded the blue dextran. We will add the buffer, then we will elute. So as we can see, the blue dextran is passing down. So we have to replenish buffer continuously, and we start collecting the eluted volume. So after complete elution of the blue dextran, we have to measure the volume, and that volume gives us void volume. Now the blue dextran completely eluted. It is around 18 to 19 ml. So now we got void volume. Now what about total volume? Total volume consists of the packed volume of the beads. Total packed volume of the beads. So it is around 25 ml of beads are there. So that means total volume is 25 ml. So with these values, after eluting the protein, suppose if you are using some unknown protein, you have to calibrate with for known proteins, then you have to construct a calibration curve between the partition coefficient, which is calculated by elution volume subtracted with void volume divided by total volume subtracted with void volume. That will give partition coefficient, and on x-axis you have to take log molecular weight. Once you plot, you will observe some correlation. Based on that, you can calculate unknown proteins molecular weight. So this is the gel filtration column attached to protein purification system. So here what we will show you, we will inject the blue dextran and BSA and show their pattern, how they are eluting. This blue dextran gives wide volume of the column, and also BSA gives actual elution pattern. So if you run few more proteins with known molecular weight, we will get the calibration curve. With that we can calculate unknown proteins molecular weight. So this column is equilibrated, as we can see here. When we introduced it into buffer, after removing 20% ethanol and water also, so we can see this one corresponds to, blue line corresponds to 218 nanometer, which is relevant to protein 1. So we can see there is a initial spike, but gradually the curve flattened. So that means there is no contaminants, and now the column is ready to inject the protein. So what we will do, we will inject the protein, and we will show how to inject protein also. Then we will show the pattern how they are eluting. So here we will end the program. So we will start the new program. system flow will keep 0.5 ml per minute. Insert flow path, column position at 1 and downward flow. Insert monitors. We need 3 different wavelengths: 215 for peptide bond, 254 for nucleic acid, and 280 for aromatic amino acids. And we have to set the alarms also. We'll set this 3 and this one 01. Complete system pressure 3 and this one 01. So we will inject the protein now, then we will see how it works. This is the port where we are going to inject the sample. So once we will inject this one, and execute, inject. So this is a pattern of injected components. This one corresponds to the blue dextran, and it gives the void volume of 8 ml. As we can see here, it corresponds to 8 ml. So there is no proper resolution between BSA and the blue dextran. This corresponds to 8 ml, which is blue dextran, and this one is the 9.2 to 9.5. This corresponds to the BSA. So once this is finished, we have to run another one column of in buffer to remove any other proteins, and after that we will keep it in water, so to remove any kind of salts if present. Then we will keep in the 20% ethanol. We will run at least one column volume, so that directly we can use to preserve the column. After that we have to purge with the 20% ethanol complete system, so that there is no contamination or bacterial growth if you left for few days also. So this is all about gel filtration chromatography. So we will show you how to analyze the result. So once the gel filtration run is over, we have to analyze the results. So this is the software we will use for the evaluation purpose. So we have to open the chromatogram which you want to analyze. So we already opened. This is the chromatogram we run recently. So we have to analyze peaks. So peak integrate option is there. So just say which one you want to analyze, UV 280 nanometer one or 215. We have only 281, so that's, let's say, analyze. So as we can see, it gave the retention volumes of the peaks and also the area and height of the peak. These values can be used for constructing calibration curve. This one belongs to blue dextran and this one is for BSA. So in a summary, in this video we showed how to run a gel filtration. We showed manually how to pack the column with the beads, and also connecting through instrument. So hope this will help for your research, to improve your research.
Now, let us see the application of gel filtration chromatography. Apart from purification, the gel filtration chromatography is being extensively used for determination of molecular weight. As you remember, we have just discussed that the molecular weight is related to the number of residues which this particular protein is going to have, and the number of residues are also directly related to the radius of gyration, which means the radius of gyration is directly proportional to the molecular weight. This is the formula. The molecular weight and radius of gyration is reversibly related, which means Rg is directly proportional to the Ma. So, Rg is directly proportional to Ma, and where A is a constant which depends on the shape of the molecule. A is 1 for rod, A is 0.5 for coils, and A is 0.33 for spherical submolecules, which means if you plot a graph between radius of gyration versus the molecular weight, and radius of gyration is directly proportional to the elution volume or VE. This means the VE is going to be inversely proportional to the molecular weight. So, to determine a molecular weight, what you are going to do is you can calibrate this column simply by running the proteins of different molecular weight, and then you plot this. So, that will give you a calibration curve between the log molecular weight and the distribution coefficient Kd of all these individual proteins which you are using for calibration, and all these proteins are globular in nature. That is why they are going to follow the basic principle of gel filtration. Then what you do is, you suppose you do the same exercise and calculate the VE, and then you calculate the Kd value of your unknown protein. Suppose this is your unknown protein, that actually will give you the log molecular weight, and then you can calculate from this log molecular weight, you can be able to calculate the molecular weight of that particular protein. Now, in this particular experiments or in this particular type of exercise, what you do not know is about the void volume. So, the void volume, how to calculate the void volume? Now, as we said, for detecting the air channels, you have to have a molecule which actually has a Kd value equal to 1, whereas to calculate the VO, which means the void volume, you have to have a molecule which is the Kd value of 0, which means in that case only elution volume is equivalent to the void volume. How to get a molecule which has a Kd value equal to 0? Now, to answer this question, you can understand that most of these gel filtration chromatography columns are only working with the globular proteins, which means they are not going to work with the fibrous proteins. So, there are two different types of protein. One is globular protein, which are ball like proteins, which actually maintains all the residues along with the center, whereas there are fibrous proteins which are actually not following this particular rule. One of the classical example of globular protein is hemoglobin. The hemoglobin, the molecule which actually carry the oxygen from one part of the body to another part. The classical example of the fibrous protein is the hair which we have, and the purpose of the globular protein and the fibrous proteins are very different. The globular proteins are mostly the protein which are actually taking part in the enzymatic reactions or all sort of metabolic reactions, whereas most of the fibrous proteins are part of the structural proteins such as the collagen or the fibrous keratin or all other protein which are actually forming this, like the protein which is making the nails. So, if you take any protein which is of fibrous in nature, which what is mean by fibrous in nature is that it is going to be of an extended conformation. Suppose you take your hair, the hair are of fiber like structure, that is why the name is known as the fibrous protein. So, if you take a structure which is of fibrous in nature, that fibrous structure is not going to enter into this particular bead irrespective of the size. So, one of the easiest fibrous structure is, instead of taking it protein, what you can do is you can take a sugar chain, you can take a sugar polymer, and sugar polymers are also present in the extended conformation. So, they will not going to enter into the beads. One of the classical example of the sugar what people use is the dextran. So, you can use the dextran, and because you want to detect this particular dextran, what people do is they just stain this particular dextran with a blue dye, and that is why the name is saying is blue dextran. So, if you load the blue dextran onto the column, the blue dextran is going to be excluded from the column because it is in the extended conformation, and it will exhibit the Kd value which is equivalent to 0, which means the VE is going to be your VO, and in that case you can just get the elution volume of the blue dextran, and that is going to be the wide volume of this particular column under that particular condition, which means if you change the pH, if you change the temperature, if you change the packaging, both for wide volume as well as the Kd values, everything will going to be changed. So, all these things are relative, whether it is wide volume or Kd values. So, as soon as you change the packaging and all other conditions, all these parameters are going to be changed, which means the calibration curve is also not going to be hold for that particular column. You also have to draw the again new calibration curves if you would like to determine the molecular weight. So, as long as the column is maintaining the particular type of packaging, you can keep using this calibration curve, but if you change the conditions, you have to redraw the calibration curves to precisely calculate the molecular weights. Now, once you calculated the molecular weight, you could also be able to calculate the oligomeric status of the protein. What is mean by oligomeric status is that either it is one monomer, which means one particular chain is going to be part of the protein, or the two chain are going to be attached to each other. So, in this case the oligomeric status is monomeric, whereas in this case the oligomeric status could be the homodimeric status or the heterodimeric status, which means it could be either NN or MM, or it could be NM. So, this is the heterodimer, these two are called as the homodimer. Similar, it could be tetramer, it could be pentamer. So, how many subunits are associating with each other and giving you the final protein is called as the oligomeric status of that particular protein. So, once you calculate the molecular weight of these protein utilizing the method just now what we have discussed, then what you can do is you can also calculate the molecular weight using the SDS page. So, SDS page is going to give you the denaturating conditions, which means in the case of the monomeric protein, it will give you the molecular weight of N, whereas the gel filtration is going to give you the molecular weight of NN, which means if you divide the NN from the N, it will say that the oligomeric status is 2, which means dimer. So, the molecular weight which you are getting from the gel filtration, if you divide that by the molecular weight which you are getting from the SDS page, that will give you the oligomeric status. Now, the next is you can study the protein folding by the gel filtration. You know that once it is generated as a small extended conformation, it gets into the native conformation, which gets folded and then it rearranges all the amino acids. If you put them under the denaturating conditions such as the urea, so if you put it under 2 molar urea, it is going to be a partially folded protein. If you put it under the 4 molar urea, it is going to be unfolded further, but if you put it under the 8 molar urea, it is going to be completely unfolded, which means this is going to be the extended conformation. This is going to be extended, which means this will behave like a fibrous protein, this is going to be behave like a globular protein, and we are only talking about the globular protein. So, the protein structure has the multi-level organization, primary structure which is the sequence of the protein, secondary structure which means alpha helix and beta sheets and turn, and the tertiary and the quaternary structure. When the protein is incubated with the increasing concentration of denaturating agents such as urea or GTMCl, it unfolds the native structure into the unfolded extended conformation following the different types of steps. Sometimes the proteins are getting unfolded completely, sometimes they are getting unfolded domain wise. So, that is why there is no universal law that the protein unfolding is going to follow a particular pattern. So once if you would like to study that, the protein is incubated with the different concentration of urea, which is from 0 to 8 molar for 8 to 10 hours at 37 degrees Celsius. A gel filtration column, if you would like to use, gel filtration column is also incubated with the buffer containing the urea, which is of same concentration, which means for this particular concentration you are going to run the gel filtration with buffer which contains the 2 molar urea. In this one it is going to contain the 4 molar urea. So, the sample also you are going to prepare in this particular denaturing conditions, the column is also going to run in the same buffer. And then mixture is analyzed. As the concentration of the denaturing agent is increasing, the protein will unfold with the increase in hydrodynamic surface area, which means if the protein is in a native conformation, it is going to make a compact ball. As soon as you are putting under the denaturing conditions, it is getting unfolded, and that is why that the diameter of that particular ball is increasing, and ultimately is going to form a extended conformation. As a result, the protein peak shifts towards the left, which means this is the peak for the native conformations, and as the size of the peak will go, the protein will come out more and more onto the left side. At highest concentration of the denaturant, the protein unfolds completely and mostly appear in the void volume. That is what we said. This is a extended conformation present just like as a fibrous protein. So, this particular protein is going to be come out into the void volume, whereas these two are the intermediate stages which are corresponding to the partially folded proteins, and this is the native proteins. Now, you can also study the protein ligand interactions. So, ligand binding to the protein induces conformational changes, results into the change in size or shape. In addition, ligand is small in size. So, the ligand, if you analyze the ligands, it is going to elute at a different size. If you study the proteins, it is also going to elute at a different size. So, the ligand which is small in size, the protein which is also small in size, if you put them together and if they make a complex, that complex is going to be of large hydrodynamic surface area, whereas the protein ligand complex is big and may appear at a distinct place in the column. So, in the step 1, what you do is a gel filtration column is equilibrated with the buffer and the elution profile of ligand is recorded. So, in the first step what you do is you equilibrate the column and then you study the elution of the ligand only. Then what you do is now column is equilibrated with the buffer containing the ligand molecules, and you can use the different concentration of the ligand. So, as the concentration of the ligand is increased, the protein binds to these ligands and they are going to start forming the complexes of different sizes, and with the increase, so they will form the complexes with the increase in hydrodynamic surface area. As a result, the protein peaks shift towards the left. So, you can see when you are analyzing the ligand, it is making a separate peak, but when you are resolving the proteins onto a column which also contains the ligands, the protein is forming a complex with the ligand, and that is how the ligand peak is shifting towards the protein side. And you can actually calculate the dissociation constant simply because after some time it is not going to migrate, and that is the place where it is going to saturate. So, the number of ligand molecules which are going to bind this particular protein is fixed. So, suppose you are resolving a 1 micromolar of protein. So, it and the stoichiometry of interaction between the protein and ligand is 1:10, then it is going to interact up to 10 micromolar with the ligand. But once you increase that particular concentration and you go to 20 micromolar, 30 micromolar or 50 micromolar, the remaining amount is going to be form the separate peaks as a ligand. So, in that case when you resolve that, what will happen is the protein ligand complex is going to appear, and then you will see that the ligand is reappearing in that particular condition. That means that now this is the stage at which the protein is fully saturated with the ligand, and if you use these particular values, you could be able to calculate the dissociation constant of the ligand protein interactions. So, as the concentration will increase with a fixed amount of the protein, the free ligand will appear in the chromatogram. The protein amount and the concentration at which the free ligand appeared and the dilution data can be used to calculate the stoichiometric ratio of ligand protein and the equilibrium constant, which means you can be able to calculate the dissociation constant K d. The protein you can also use for protein desalting. So, you know that sometime the protein is being purified by for example, if you are purifying a protein using the ion exchange chromatography, you are actually putting lot of salt, and that salt is you are using for elutions. Now, if in a subsequent step, suppose you would like to do the enzymatic reaction and that salt is going to interfere, or suppose you want to use this protein for any other application and this salt is going to interfere into your analysis, then you have to remove this salt, and there are multiple ways in which you can remove this salt. One of the way is that you can put this dialysis of this particular protein against the buffer. If you do a dialysis, the salt is going to be exchanged along with the water, and that is how the water will come in and the salt is going to be removed. But the dialysis is a very small step. So, in case some proteins are prone for degradation or some proteins are getting inactivated, if you are going to continue this process for 10 hours or 12 hours, in those cases we are also using the gel filtration chromatography to remove the salt, because salt is nothing but the small molecules compared to the protein which is of large molecules. So, if you inject the protein onto a gel filtration column, the protein is going to form a separate peak and the salt is going to form the separate peaks. So, because salt is small in size, it will going to come and it will sit into the bottom of the beads, whereas the protein will go and bind to the other sides. So, because of that, a gel filtration column is equilibrated with the buffer or water, and then the sample for desalting is loaded. After the run, the protein and salts are elute separately as a peak. So, if you elute, the protein will come on first and the salt is going to come the later on. So, you can collect these proteins amount, and you can be that is how your protein is going to be separated from the salt, and this protein can be used for downstream applications. So, now with this we have already discussed about the ion exchange chromatography, we have discussed about hydrophobic interaction chromatography, and we have discussed about the gel filtration chromatography. And so far we have discussed about all these three chromatography techniques which people are very oftenly use for protein purification as well as the characterization of the proteins. In general, people are running the ion exchange chromatography first, followed by the hydrophobic interaction chromatography, and followed by the gel filtration chromatography. The reason is that ion exchange chromatography requires the protein to present without salt, whereas the hydrophobic interaction chromatography requires the high salt concentration. So, the fraction which you elute from the ion exchange chromatography already contains salt. This means you can directly load those fractions without going through a process of dialysis or desalting into the hydrophobic interaction chromatography, and once the sample which you are going to get from the hydrophobic interaction chromatography is going to be of low salt, because if you remember, the elution conditions for the hydrophobic interaction chromatography is that we are decreasing the salt. So, your sample or the fraction which actually will contain your protein are going to have the low salt conditions, and then you can directly load those low salt proteins onto the gel filtration column to purify or to characterize. Now, in the subsequent lecture, we are going to discuss about the affinity chromatography, and we are going to discuss about how to generate the affinity columns, and we are going to take up different examples of affinity chromatography. So, with this I would like to conclude our lecture here. In the subsequent lecture, we are going to discuss about the affinity chromatography. Thank you.